But first we have to back up a step. We have to think about contraction mechanism. So let's look at the contraction proteins. I should have said when I hit back up a step, this is skeletal muscle part 2. So you are in the second one if you hit the back up a step slide. So we're going to look at these contraction proteins. The first protein is called myosin. Now you may have heard of myosin and actin before. We're going to look at them. I took this picture from the book because it's a little bit more neat than I can draw it. We have this long twisted tail with these 2 active head groups. Now on the head group, we have an actin binding site. It wants to bind the protein actin. And we have an ATP ACE. Remember, ACE equals enzyme. Any enzyme called an ATP ACE specifically burns ATP for the energy. And we're using it much like the pump, burn it to move the sodium. This time we're going to move the myosin and the actin. So normally what we see is that these myosin tails are twisted and then they're end to each other. And then we kind of bundle them up in a whole group where each one of these pairs is one of this pair of head groups. So we see that the bare zone is because this is in the center of the sarcomere. And what we're going to see is this bare zone actually has a protein called the M line in it. And this is how we're going to do this. Now, myosin is sometimes called the thick filament because it's thicker and denser than the actin, which is why if you remember looking at a picture of skeletal muscle, or you remember looking at the skeletal muscle cells in the microscope from anatomy, or the photograph of the slide, you'll remember that it was striped. It had those striations. Those striations, the dark region is where the myosin was. Now let's think about actin. Actin's the thin filament. And we start out as this globular protein called G-actin. G stands for globular. And what we see is that it's just a little round circle and they have these little myosin binding sites. But then we put it together. F stands for filament. So we make a filament of actin and then we twist it together as a double helix. Now, if you want to think about how this would work, think about beads on a string and you just twist them together to work to wrap them around each other. Now, we need 2 more proteins to make this all work. We have the regulatory proteins because you don't want your muscles to be constantly contracting. They need to relax. We need to be able to control it. So to control muscle, we have tropomyosin and troponin. The key is tropomyosin wraps around the actin, and you'll see here it's blocking all of these little myosin binding sites. So myosin cannot bind to actin if tropomyosin is laying over the top. And then we have troponin. Now, troponin's job is to regulate the tropomyosin, because if I change the troponin, it will change the tropomyosin. So here I've drawn, or I took the picture of the sarcomere and I drew in the M line. So what we have is we have the actin, these thin filaments that overlap the myosin. This is kind of squished in a little bit. The actin is all connected by something called the Z line. You can see Z stands for zigzag. So we have Z line. Now one sarcomere is Z line to Z line. And then we have the myosin held in the center, and it's the thick filament. This is another picture from the book, because remember when you looked at the skeletal muscle cell, those stripes, each stripe, dark stripe, was the myosin. So the dark light was where the actin is. And so I wanted to show that this thing is repeated down the cell over and over and over again, thousands of times down a single cell. And what we see is that while we usually draw it as if it's flat, it's actually arranged so that the actin is in a circle around the myosin, and each myosin is surrounded by actin. So to actually contract, we need something called the sliding filament mechanism. So this is actual contraction. So when we think about this sarcomere, contraction is we are going to shorten the sarcomere. Now, if I shorten every sarcomere a little bit, the whole muscle shortens. Now, I want you to notice that in this picture, they specifically have the actin stays the same length. The myosin stays the same length. What changes is the distance between the two sets of actin. We're literally pulling this actin towards the middle, and the space between them is decreased. We get the best tension when we have the best overlap of actin and myosin. It's kind of like if you had somebody pulling on a rope and everybody pulls towards the center. So and that's why it's called the sliding filament, is the thin filament slides over the thick filament. Now we're going to go from sarcomere to contraction. We built our sarcomere. This is muscle part 3. So the whole question becomes how? We had the calcium flooding the cell. We have the structure of the sarcomere. How do we put all of this together to get that excitation, action potential, contraction, actual movement connected through the calcium? Here's what happens. Calcium binds the troponin, which then moves the tropomyosin off the binding site, so myosin can now attach to the actin. And here's a picture.